We're good to go.
Hello, everybody, and welcome to the QLS seminar series for today. We are really happy to have with us today Dr. Matthew Stevens from the University of Chicago. He did his PhD at Oxford University at Magdalen College with Brian Ripley and a postdoctoral fellowship with Peter Donnelly. He won the Guy Medal for the Royal Statistical Society in 2006. And many of you will probably know a lot of his work over the years. He's developed a lot of work on population structure, looking at genome-wide association studies in different ways, and more recently looking at single-cell RNA-seq work, doing imputation for genetic variants, and many, many other contributions in all kinds of areas of genetic and genomic statistics. So it's a real honor to have him with us today. He has said that he'd welcome questions during the talk. So I'll keep my eyes on the chat window, and you can put your questions there. And otherwise, I'm very happy to hand it over to you, Matthew.
Well, thank you very much, Celia. Thank you for having me. It's a pleasure to be here. Yes, and Celia said if there are any questions along the way, please just raise your hand or put it in the chat. I probably won't see the chat, is my experience, but if you raise your hand, I'll probably see you. Or just unmute yourself and interrupt. Happy to take questions along the way. So, okay, so this is work that was done with a great team of postdoctoral researchers, students, and scientists. So Gao Wang is now at Columbia. Peter Carbonetto is a computational scientist in my lab. Abhishek Sarkar was a postdoc with me, and Yuqin Zhu was a graduate student. They've both just started new jobs in industry. Yeah, let me, let's see if I can, yes. Okay, so we were kind of motivated by this work. We're motivated in this work by our involvement in the GTEx project, so the Genotype Tissue Expression Project, where what the Genotype Tissue Expression Project, the GTEx project, did was measure gene expression in lots of different human tissues in lots of different people. So it doesn't really, for this, on the y-axis here, we've got different tissues. You don't really need to know what they are. Some of the codes are a bit obtuse, but you can see there's breast, there's lung, there's nerves, there's different skin types. So that's what each row here is a tissue. And then, so they measured gene expression in lots of people in those tissues, and then they also measured genetic variants in the same people, so SNPs, essentially. And one of the goals was to identify genetic variants that were involved in regulation by identifying genetic variants that are associated with gene expression in different tissues. So these are so-called eQTLs, or expression QTLs. So for each gene in each tissue, the kind of default routine analysis is to just compute a measure of association between that genetic variant and between each genetic variant, with expression in that gene in that tissue. And usually those tests are done, at least initially, those measures of association are computed for SNPs that are nearby the gene that you're looking at, because most of the strong signals tend to be nearby. There is a lot of interest in identifying more distal signals, but for now, let's just look at these more proximal signals, sometimes called cis eQTLs. And so what's shown here is for each tissue, for each SNP, we've computed a measure of association between that SNP and gene expression in that tissue for a specific gene. Okay, and these bars, the height of the bar, or the color of the bar shows the effect size, and I think the height shows maybe a p-value, which of course is kind of they're kind of highly correlated. The bigger effect sizes, the stronger associations tend to have smaller p-values, and so bigger bar. Okay, so what you see in this picture is a lot of bright colors. Blue for negative effects, red for positive effects. And so one of the things, okay, so that just means there's a lot of genetic variants that are associated with gene expression in these tissues. And one of the things that people will use this kind of data, this essentially forms a database. And one of the things people will use this database for is to go and look up for a particular SNP that they're interested in. Maybe they found it's associated with some disease and they want to know, oh, is this SNP an EQTL in the tissue I'm interested in? Okay, there was a question. Let me just... Was this bulk RNA? Yeah, this was bulk RNA sequencing, yes. And so they asked the question, is my SNP that I'm interested in an EQTL in a relevant tissue type maybe? And if they just go and look here, the answer is almost certainly going to be yes. Because almost everything is associated with gene expression. At least at this, in this picture. Well, maybe almost everything is a bit of an exaggeration, but you can see there's a lot of red here. But we don't really believe that all these genetic variants are actually doing something to gene expression. What we believe is that these many of these associations are essentially due to what's called linkage disequilibrium between the SNPs, or correlation between the SNPs. That is, maybe there's one or a small number of SNPs that are actually doing something, that is causing a signal of association. They're actually maybe influencing the gene expression. And then the other SNPs, because they're correlated with those let's call them causal SNPs, because they're correlated with the causal SNPs, they're also correlated with gene expression, so that's what's driving these associations. So you see, the problem we have is that even one SNP doing something causes lots of SNPs to be associated. And what we'd like to do, so the goal of what's sometimes called fine mapping is to take data like these and narrow it down to narrow down what are the SNPs, hopefully a small number of SNPs, that might actually be responsible, the causal SNPs that might be responsible or explain the associations we're seeing across lots of SNPs, right? So how many SNPs do we need to explain these patterns of association? How many causal SNPs do we need to explain these patterns of association? So we'd like to do this ideally by looking across all the tissues simultaneously. And the work I'm going to tell you about was kind of motivated by me trying to write a grant proposal saying about how we were going to do that, and scratching my head and thinking this is a really complicated problem. And actually methods for even if we look at one tissue, the existing methods are really quite complicated. In some ways they work quite well, but they're already quite complicated. And writing a grant proposal for how we were going to extend these quite complicated methods from one tissue to, say, deal with 50 tissues was a bit overwhelming for me. I couldn't even convince myself that we were going to be able to do it. And so the first step I felt was we have to simplify. We have to simplify the single tissue case, the univariate, what you might call the univariate or single outcome case first. And so that's what I'm going to tell you about. We're now in the process of extending that single tissue case to deal with the multiple tissues, but I'm only going to tell you about the single tissue case right now. Okay, so that's what this green here. Let's focus on this. So in this single tissue, we've got association. We've still got association across many SNPs, and the question is which SNPs might be explaining this association. Okay. So a little bit of background and introduce notation. So association tests, the kind of results I just showed you, are usually done SNP by SNP. That is, for each SNP J, you fit, say, a linear or a logistic regression, depending on whether continuous or discrete or binary. But here, so a linear regression here Y here is going to be the phenotype, which in my example is gene expression, but it could be a complex trait in a GWAS. Xj is the genotypes, a vector of genotypes. So Y is a column vector of expression data across different individuals. Xj is their genotypes at a single SNP J, and beta J is the the regression coefficient for SNP J in this single SNP regression. And so if we want to test the null hypothesis that SNP J is unassociated with expression, we're testing the null hypothesis that beta J is 0. Okay, so beta J here purely measures an association of SNP J with Y. Right. Okay, so that's simple. Hopefully you've all seen linear regression before. You don't need to memorize these, but they're going to kind of come up. The idea is that these are very simple forms. If you've seen them before, you'll recognize them. If you haven't, don't worry about memorizing these. But essentially, there's a simple formula for the estimated beta, that's beta hat, and its standard error, and you can form a z-score by dividing the estimate of the coefficient by its standard error. Okay, so these are going to come up later, but they're very simple calculations. And this is also going to come up later, and might be something you haven't seen before, but another way of measuring instead of computing a p-value, say testing the null hypothesis, another way of measuring the significance of an association is to compute a Bayes factor. So the Bayes factor measuring the strength of the evidence for an association between SNP J is the ratio of the probability of the observed data under the model where SNP J is associated, that is under a model where beta J is non-zero, versus divided by the probability of the data under the null hypothesis where beta J is 0. So essentially you might think of this as a likelihood ratio where the top is integrating out over possible effect sizes. And under simplifying assumptions, these Bayes factors are also very easy to compute. And again, you don't need to memorize this. All you need to know is that it's a function, or all I'm trying to say here is it's a simple function of the z-score for SNP J and the standard error here. And there's another parameter here which is has to be fixed, which specifies how big an effect size we expect to see. So essentially this Bayes factor, unlike the p-value, the disadvantage of the Bayes factor is it depends on assumptions about the effect size, but the advantage of it is that it's a bit easier to interpret directly as a measure of evidence than a p-value. So when you want to interpret p-values, you have to start thinking about power, and if you want to think about power, you have to make assumptions about effect sizes. So it's kind of like, that's what's happening here is with the Bayes factor. You make a little bit of an additional assumption, which is about how big you expect the effect sizes to be, and you get a direct measure of the evidence for SNP J being associated with each Y. Okay, the formula is not important. The point is it's super easy to compute. Okay, so that was single SNP regression. So now what I want to talk about is multiple regression. So this is in matrix format. What this says is X is now a matrix of the genotypes, and B is a vector, one element for each SNP with its regression coefficient. And the difference between this, you can think of this as a sum over all SNPs of xj, bj, the previous, in the model before we had y equals xj, beta j. So we can write a sum over j here, and it's just being written as a matrix here. Okay. So this is adding up essentially all the quote effects of all the different SNPs. But I've used b instead of beta here because b is not the same as beta. Beta j was just a measure of association between SNP j and y, whereas bj is the association of SNP j with y after controlling for the effects of all the other SNPs, right? Because we're looking at them all at once, the b has a different interpretation. It's the effect of that SNP after adjusting for the effects of all the others. And so, again, under simplifying assumptions, and if we think all the SNPs have been typed, we can actually interpret Bj in this model as the causal effect of SNP-J. So unlike beta J in the SNP by SNP model, which was just a measure of association, I'm going to interpret Bj in this model as representing the causal effect of SNP-J. And you need to make some assumptions, but they're not too, I think that it's a reasonable starting point. So in other words, fine mapping just involves fitting this multiple regression model. And that's how almost everyone does fine mapping is, so if you're familiar with fine mapping, this will all be background. And if you're not familiar with fine mapping, you can now just say fine mapping. I didn't know about fine mapping, but all it is really is multiple regression. And I know, well, maybe you know a bit about multiple regression. If you don't, it doesn't matter. I'm going to tell you a bit about multiple regression. But if you do, you can fit it into that framework. And in even more, fine mapping is really about variable selection in the multiple regression. That is, for most genetic applications, we can assume that this vector B has most elements that are either 0 or very, very close to 0. That is, very small effects, but a few elements have larger effects and maybe big enough to detect. And we're looking to see which elements of B may be non-zero. So in statistics, this problem of deciding which elements of B are non-zero is sometimes called variable selection. The variables here are the SNPs, and we're trying to select which SNPs have a non-zero B. That is, we're trying to say which of them has a non-zero effect. And just to mention, in some, perhaps many applications of variable selection, the motivation for people selecting variables is to improve predictive accuracy. So there's, there are models, there are many settings where you might want to select variables in order to regularize your regression and in order to just improve prediction. But in our application, we're not trying to improve prediction. I mean, we might want to sometimes, but we're really trying to understand something about nature. We're trying to understand which SNPs are actually causing a change in gene expression. So we're really interested in saying which SNPs have a non-zero effect. And that's going to be important later, it turns out. Okay, before I go through this toy example, are there any questions? There seems to be a hand up in the room, but...
I was hoping, could you provide maybe some biological reasons why some SNPs are correlated with each other?
Oh, okay, great. Well, the reason for that is essentially that SNPs that are nearby one another on the genome tend to be inherited together from generation to generation. So that's what because they're inherited together. When a mutation arises, it arises on a particular background, and then it tends to get transmitted with that genetic background. And so that's the biological reason for why SNPs are correlated. And from what my explanation, you'll kind of impute maybe that SNPs that are nearby tend to be correlated with one another because they tend to be inherited together, whereas SNPs that are far apart, I mean, certainly on different chromosomes, but even just far away on the same chromosome, will often be uncorrelated. So the correlation structure we see in genetic variant data is very local. If you plotted a correlation map of the whole genome or a whole chromosome, you would see very strong elements just near the diagonal and quite a narrow diagonal. Maybe I'll also mention that unlike some statistical applications, these correlations are very, very strong. Sometimes they're literally 1 in your sample. That is, 2 SNPs can be completely correlated. If you've got one genetic variant at 1, you've got another with, everyone has matching genotypes at the 2 SNPs in quite large samples. That can happen, and although 1 is plausible, 0.999 is also quite common. 0.99 is common. Really high correlations, and that also actually, it turns out, matters. And it's going to come up in my toy example here because just to illustrate why this is a hard problem, because this is multiple regression. So, there's been a lot of work on this. Why do we need more work? Let me just explain why it's a hard problem and why existing methods struggle to solve it. So in my toy example here, I'm going to pretend let's say you've got 1,000 SNPs, just for concreteness, doesn't really matter. And I'm going to suppose that the SNPs 1 and 2 have exactly the same identical genotypes. So that is, the first column of X is equal to the second column of X. So they're 100% correlated. And I'm going to assume that the third and fourth SNPs are also 100% correlated. And then for simplicity, just assume that X5 to X1000 are uncorrelated. And let's suppose the truth is that SNP1 and SNP3 have an effect. So here, SNP1 has a big effect. It has a coefficient of 5 in the multiple regression. And SNP3 has a smaller effect. It has a coefficient of 1. And everything else has no effect. So it's clear from the setting that we can't distinguish between variable 1 and variable 2. They're SNP1 and SNP1. They're completely correlated. There's no statistical method that can distinguish them. So in other words, and this is common in genetic fine mapping, getting the correct answer is impossible to guarantee. We can't possibly say that it's SNP1 that has an effect because SNP2 has exactly the same genotypes as SNP1. So the best we can say is that either SNP1 or SNP2 has an effect, but I can't tell you which one because they're completely correlated. And similarly, I can say the same thing for 3 and 4. X3 and 4 are also completely correlated. They're uncorrelated with X1 and X2, I'm assuming. So you can say that also either X3 or X4 has an effect, but I can't tell you which one. Right, so the idea is that given enough data, and this being the truth, I ought to be able to tell you either SNP1 or 2 has an effect and either SNP3 or 4 has an effect, but I can't tell you whether it's 1 or 2, and I can't tell you whether it's 3 or 4. So that's the kind of inference I'd like to get out of this toy example. Another way of thinking about it is in terms of what we're going to call credible sets. So a credible set of variables is a set, I'm defining it to be a set that has a high probability, let's say 95%, but you could choose another value, to contain at least one variable that has a non-zero effect. So I'm going to give you a set of SNPs and I'm going to say at least one of these has an effect. I just can't tell you which one because they're all, presumably, because they're all correlated with one another. And we want to, of course, find lots of such credible sets because these are kind of our discoveries. If I give you a set, I'm saying one of these has an effect. I just can't tell you which. And I want the sets to be as small as possible, just like you would usually want intervals to be as small as possible while still containing the true value, or in this case, while still containing a causal SNP. So in my example, I might give you two credible sets, one containing SNPs 1 and 2, and the other containing SNP 3 and 4. So that's another way of framing what I'd like to be able to do. Provide credible sets, each one as small as possible, and each one contains at least one causal SNP, but I can't tell you which one. And the kind of point, which was true at least when we started this work, was that most existing methods don't give you that kind of credible set. So many approaches to sparse multiple regression have been developed. Some of them are based on penalized approaches. You may have heard of lasso or elastic net. And essentially what they do is just find a single solution and give it to you. They say, here's a set of variables that may explain the data. I'll take the question in a minute. And another approach, which gets close to solving the problem, but still doesn't quite give you what we've just said, is Bayesian regression via all subsets, MCMC, stochastic search. And I'll talk a little bit more about that in a minute, about why that doesn't solve the problem easily anyway, fully. Yes, question?
I was just wondering if, so in the toy example it was pairwise, but would the things you do also be pairwise, or do you take into account potential interactions or multiples?
When you say pairwise, do you mean that each set contained two, or do you mean something else?
I guess it was are you comparing, if I have a region of SNPs, am I saying this one is causal, this one is not, this one is not, while fixing X1, let's say I'm fixing X1 and then looking at everyone else one at a time? Or am I looking at them together?
What do you mean by fixing X1?
You assign a fixed value.
Do you mean B1?
Sure. I assign it a fixed value, and then I see how everything else changes.
Yeah. So for now, I'm not talking about any particular procedure, right? I'm talking about an inferential goal. That is, if these were the if I observe y and x in this model, and these were the true values of b, what might I be able to infer from that data in an ideal world? Given I'm not telling you yet how I'm going to do it. I'm telling you what my goal is.
So I guess if I look at that sentence, it would be select variables 1 or 2 and 3 or 4. So, but it would be variables 1 and 2 and 3 or 4.
Why do you say 1 and 2?
So perhaps each individually is not causal, but when they're together, they are.
I see. So I would say that inherently this is an additive model, right? If you look at the top, that it's an additive model. So we're assuming the effects are additive.
Okay.
You could potentially try thinking about putting interactions here in as variables in the model. Like you could include a column of X for the interaction term. But that, yeah, we're not, then you might want to start putting in hierarchies of only including an interaction if there's a main effect or things like this. So it would get more complicated. So yeah, we're inherently assuming additive, but not pairwise. I mean, going back to your question, we're not assuming, there's nothing about 2 here. We could have said, I could have had 4 variables that were completely correlated with one another. And then my inference would have been, it's one of these 4, but I can't tell you which one. Right?
Thank you.
Good. I think there was a chat. Is a linear model something that is reasonable to assume in practice? Yes. In genetic association studies, I would say, particularly with complex traits, the vast majority of analyses assume additivity, and that's been reasonably successful, and people haven't been that successful at finding interactions. That may not imply that they don't exist. It may just be that they're harder to find because there's lots of them, etc. It's kind of, it remains a something of a question to what extent quotes missing heritability may be due to interactions. But it's a common and common and useful assumption. Yeah, I would say. Any other questions? I'll just mention, there are ideas that people have done. So one idea here is maybe we should cluster the variables first and then select clusters instead of selecting variables. So in my example, we could have clustered variable 1 and 2 together and called it one variable, and then when we select it, we would know it was either 1 or 2, but we wouldn't know which. So that's an approach that people have taken. And the problem with that is that cluster, it's fine for my toy example, but in practice you just don't know the variables do not fall into nice discrete clusters of highly correlated variables and then another set that are very highly correlated with no correlations between them. So the problem is you don't really know how to cluster the variables in advance. In my toy example, you kind of do, because I've shown you 1 and 2 together, 3 and 4 together. But so what we're going to do is actually try to avoid pre-clustering the variables, but you can think of what we're doing as post-clustering in a way. It's like using the Y to say which variables might have an effect, but we are so correlated we can't distinguish between them. So why do the Bayesian approaches, or how do the Bayesian approaches work, and why do they not solve the problem? So the way the Bayesian approaches work is essentially to enumerate either, conceptually they consider all possible combinations of which variables could be included and compute a measure of support for how well the data match that set of variables being included. And then they also have a prior distribution that says they expect things to be sparse. So let me just, in my toy example, there are 4 variables that are actually kind of interest. The rest, given enough data, just don't have any effect, so they don't come into it. And essentially what the Bayesian method would do is say, there's one model where SNP 1 and 3 is included. That's actually the correct model in this case. But there's another model where SNP 1 and 4 are included, and that model is actually the same as the true model because SNP 1 and SNP 4 and SNP 3 are identical. And there's another model where 2 and 3 are included, and that's also identical to the true model because SNP 2 is the same as SNP 1 and SNP 3 is the same. And then the last one is 2 and 4, and this is also identical to the true model because SNP 2 is the same as SNP 1 and 4 is the same as 3, so it's the same as this. So these 4 models are indistinguishable from one another. They all kind of explain the data equally well. They're all equally sparse. They all contain only 2 causal variables. So they're under a sparse prior, they'll typically have the same prior support as well. And so the result is that a Bayesian method would end up assigning 25% probability to each of these 4 models. Right? So it would say, it's one of these 4, I can't tell you which one. Which is kind of getting close to what I said, but not quite. In fact, this contains all the information that you need to say. If you stare at it a while, you'll realize that what this is really saying is it's either SNP 1 or 2, and I can't tell you which. And it's either 3 or 4, but I can't tell you which. That logical statement, either 1 or 2 and either 3 or 4, is actually kind of equivalent to saying it's one of these 4, but I can't tell you which. So if you stare at it, you realize you could extract the information I want to extract from here. But it's not trivial to do that. And as you can imagine, this is a toy example. In practice, what the Bayesian methods do is spit out 100 different models, each with some tiny probability, and say, here are all the possibilities. Each one has a weight. And many of them will contain many SNPs. So you can't boil it down to like a simple statement of, well, it's either one of these 2 and I can't tell you which, and it's either one of these 2 and I can't tell you which. So one way of thinking about this is it's a challenge of summarizing the posterior. And I'll take the question in a minute. And so what do people do? Instead, they summarize the posterior by just looking SNP by SNP. So for each SNP, they say, what's the probability that this SNP is included? And if we look at SNP 1 here, the probability it's included is these 2 models, which sum to 0.5. So SNP 1 has a 50% chance of being included. And so does SNP 2, 50%. And so does SNP 3. If you add up the models containing SNP 3, 50%. And so does SNP 4. So the standard thing to do would be to compute for each SNP here what's called a posterior inclusion probability. It's the probability it's included. And in this example, it would be 50%, 50%, 50%, 50%, which has lost the information that it's either 1 or 2 and either 3 or 4, right? It's kind of lost that information by just looking marginally at each SNP. Okay, it's a question. How many credible sets would you expect here? 4? No, that's more. I would hope for 2 credible sets, because the idea is ideally we want a credible set for each real effect. Here there are 2 real effects, and we'd like the credible set to be as small as possible whilst containing the real effect. So here there are 2 effects. We'd hope there was a credible set for each one. So ideally, the number of credible sets will be the same as the number of causal SNPs. We're also going to be, I guess, focusing on trying to avoid false positives. So maybe we would say the number of credible sets is maybe a lower bound on the number of causal SNPs. In fact, it could be that every SNP is doing something, right? just many of them are having an effect so small we can't detect it. So so the way I think of it is the number of credible sets is kind of a lower bound on how many SNPs we think there will be. The true effects would be in disjoint CSs. Ideally, yes. Yeah, if they're distinguishable. Right. So if two SNPs are completely correlated with one another and both have an effect, we can't distinguish them and we'd put them in the same CS. Right. But ideally, we'd like them in different CSs. But if they're correlated with one, we can't do that. We have to put them in the same. They're indistinguishable. And all we can say is at least one of these has an effect. So it could be both of them, but you know, sparsity dictates that a parsimonious explanation would be only one of them. But we don't claim that only one of them in a CS has an effect, because you know it's possible everything has an effect. So ideally, yes, we've got as many CSs as effects and as small as possible, each containing a single effect. But we don't require that. And we can't. I mean, we just can't. A kind of important principle overall is you can never be sure that something doesn't have an effect. So if something is not included in a CS, that doesn't imply it doesn't have an effect. Just like if you don't reject the null, it doesn't imply the null is true. Okay. So some of the challenges with existing Bayesian approaches, the computations are time consuming to run and also to code. They're quite complex. And also going from the output of like a list of 100 models and their posterior probabilities, let's say, or maybe if you're doing MCMC, a sample from the posterior distribution of all possible models, going from that list or that output to a simple set of inference, like, for example, the credible sets I've just described, it's tricky. It's got like a statistical inference problem in its own, is going from that posterior to something you can actually interpret. And these posterior inclusion probabilities that people are computing routinely, including us, they don't contain all the information. So this is how we set about trying to solve the problem. So first thing, simplify. Simplify to a case that we can solve. So it's a super simple assumption. We're going to have to relax it later. But for now, let's make this super simple assumption that there's exactly one SNP that has a non-zero effect. I'm going to call this the single effect regression. So if we assume it's a multiple regression model, but where the multiple regression, the vector, has only one non-zero element. So only one SNP has an effect. So it turns out that if you want to fit this model, this single effect regression model, it's super easy because there are only p possibilities. If there's only one SNP that has an effect, it could be SNP1, it could be SNP2, it could be SNP3, it could be SNP4, up to p, where p is the number of SNPs. So if you've got 1,000 SNPs, there are only 1,000 possibilities. So the way you fit this model is just to enumerate all 1,000 possibilities. And it turns out that when you do that, the posterior inclusion probability is now sufficient. It's exactly what you want because it's the probability that it's that SNP and not another one. And it's given by this simple formula here. It's the Bayes factor divided by the sum of the Bayes factors so that they sum to 1. So the Bayes factor here is just that single SNP Bayes factor that I introduced at the start based on the single SNP regression. So basically, just to say what you're doing, you're computing a Bayes factor for each SNP. Say there's 1,000 SNPs, you compute 1,000 Bayes factors, and then you normalize them to sum to 1, and those are the posterior inclusion probabilities, which say what's the probability it's that SNP. And we can get away with that because there's only one SNP under our assumption. Right? So this seemingly ridiculously simple model has actually been quite widely used in fine mapping. It's quite useful because although it might not be true there's only one SNP in all regions, in many regions actually it might be enough to explain the data. Right? So so you could look at, for example, at Mauler 2012, who used this approach to try to do fine mapping under the single effect regression, the single causal SNP assumption. Okay. And and the point here is if you want a CS, you just you rank the SNPs in order of their pips, and you just take the biggest pip and you keep adding the next biggest pip until you get to 95% or whatever value you want. And that gives you a set that has 95% or whatever value you want probability of containing the, because there's only one, causal SNP. So that's what Moller et al. Pointed out. So how does that work in my toy example? Well, okay, my toy example had 2 causal SNPs, so let's simplify it to make 1 causal SNP. So I'm going to take away the second causal SNP for now, just for illustration. How does it work in the case when there really is 1 causal SNP? Well, what will happen is that it's going to compute a Bayes factor for each of the SNPs. SNP 1 has an effect, it's going to have a big Bayes factor. SNP 2 doesn't have an effect, but it's completely correlated with SNP 1, so it's going to have the same Bayes factor. So they're going to have 2 big Bayes factors, and then all the other SNPs are uncorrelated with x1 and x2, I'm going to assume in my toy example, so they're going to have small Bayes factors, they're not going to be associated, so you're going to have 2 big Bayes factors, and that's going to lead to 2 pips close to, say, 0.5, all the other pips are going to be very small, and my credible set is going to contain, well, I order these and add them up until they get to 95%, and I get to include both SNPs 1 and 2. I add SNP 1, then I need SNP 2 to get above 50%, gets to 100%, and I say, okay, that credible set now has very high probability of containing the causal SNP. So in other words, in my toy example where there was exactly 1 causal SNP, the single effect regression gives me exactly the right answer. So, okay, so what we want to do, obviously, is to generalize this single effect regression to allow for more than 1 effect. And the usual approach people have taken to do that is to simply allow that more than one of these BJs be non-zero. But that causes an explosion of the number of possibilities, and the computation becomes extremely challenging, and you have to resort to these MCMC schemes, etc. So our new approach is instead to add up multiple single effects. So what do I mean by that? Okay, so instead of writing B and saying B can have a vector with some non-zero elements, I'm going to write B as a sum of these vectors. So each BL here is a vector of length P, and each BL has exactly 1 non-zero effect. And there's going to be capital L of them, which means when I add them up, element-wise addition, so I'm adding up a set of vectors, each vector has 1 non-zero element, so their sum will have L non-zero elements or fewer, at most L, because they could have the same non-zero elements sometimes. That'd be unlikely if P is big and L is small, but it's not impossible. Okay, so we've just essentially we've reparameterized the model, and we call this the sum of single effects model, or SuSiE. So B here is a sum of vectors. Each vector has 1 non-zero element, so that's a single effect vector. So this is a sum of single effect vectors. And it allows for up to L non-zero effects. So just for now, let's just think of L as 10, let's say. So we're going to allow for up to 10 non-zero effects, which is already much better than 1 non-zero effect. But that's in EQTL analyses, 10 is a lot of effects. So, that's already going to cover the vast majority of genes. Okay, there's a question in the chat. L come from pip? No. L here is a user-defined parameter of the model. I'm just going to say, let's just think of L as 10. I'm going to say more about L later. For now, let's just say L's 10. I'm going to allow for 10 effects, and we'll come back to that. Okay, so why is this a good idea? Why does that help? Why does this help? So the key point is that if we assume temporarily that we know L minus 1 of these vectors, and we only want to estimate 1 of them, then it becomes a single effect regression. Essentially, because we know L of them, we can kind of subtract them out of Y, take them to the left-hand side, so that the right-hand side only contains one BL that we don't know. I guess this should be a capital L here, right? If we know all of them up to L minus 1, then fitting the Lth one, it's just one single effect regression again. So the idea is that this parameterization suggests an iterative algorithm. The iterative algorithm is one where you iteratively estimate the BLs. Each BL is a single effect vector, so you run a single effect regression to estimate the PIPs for each SNP, but now these PIPs are not the probability that that SNP is the causal effect in BL, and there's going to be another set of PIPs. I guess I shouldn't really call them PIPs, but another set of probabilities for each of the L effects is going to have a set of probabilities saying what's the probability that that SNP has the non-zero effect in that vector. So the idea is that this parameterization is useful or helpful because now we can just iterate from L equals 1 to capital L and go back to fitting single effect regressions, which we know how to do. So I want to draw an analogy. If that didn't quite, if you're a bit confused, let's draw an analogy with a simple routine called stepwise selection. Actually, let me see the chat. In the single effect regression model, if a non-causal SNP has the highest marginal Bayes factor, will it give the highest pip to that SNP? The answer is yes. That the highest marginal Bayes factor will necessarily have the highest pip. Perhaps a more realistic model is to consider how the LD of each SNP correlates with the Bayes factors of all the other SNPs. Well, that's the beauty actually of the single effect regression, is that under the single effect regression model, the LD between SNPs actually becomes irrelevant. Under the single effect, it's a mathematical fact that you don't need to know the LD. All you need to know is the Bayes factors. And of course, if the LD between two SNPs is very strong, they will have similar Bayes factors. But you don't need, all you need is the Bayes factors. If I tell you all the Bayes factors, the single SNP results, you no longer need to know the LD in order to do inference. That's only true for the single effect regression. So it's one of the reasons that single effect regression model is super convenient, but also overly simplistic. So, I kind of get your intuition is maybe you want to try and do something taking the LD into account, but it turns out you don't need to. Under the single effect model, the LD becomes irrelevant. That's not true under the sum of single effects regression model, where you allow for more than one SNP. We need the LD. And we account for it. Essentially, when we found one effect, we have to control for that effect when finding another effect. And controlling for that requires knowledge of the LD. Okay. I need to get a move on to this. Thank you. Okay. So let me just go through this analogy. So stepwise selection, if you haven't heard of it, has this simple form. For each SNP, you would fit this univariate model and you would choose the SNP that has the strongest association. So you just test each SNP for association with Y, choose the SNP with the strongest association, remove its effect, and repeat. Look for another one. Right, that's what stepwise selection does. So actually, our algorithm is a little bit like stepwise selection, except instead of choosing the SNP that has the best fit at each case, we average over the SNPs that have similarly good fits. So we essentially compute a Bayes factor for each SNP, and then we compute a weight for each SNP, which you can think of, instead of selecting a single SNP, we select every SNP but with a weight. So maybe one SNP has all the weight, and that would be just like the stepwise selection. Maybe two SNPs have 50-50 weight. That would be a bit different from the traditional stepwise selection because you'd be kind of selecting both of them 50-50. Or maybe every SNP has a very small weight, and that would be completely different from stepwise selection because we'd be selecting everything but with a tiny weight. But that's essentially our algorithm is an iterative version of this kind of, you might call it fuzzy stepwise selection. So let's see how this works in our toy example. So if we did forward selection at the first step, because the first SNP has the strongest effect here, it's 5, either 1 or 2 would be selected, but we wouldn't know which one. So we'd select one of them, but it'd be arbitrary which one. And then we would look for a second SNP, and then either 3 or 4 would be selected, but it would be arbitrary which one. Whereas in our procedure, at the first step, we notice that we can't tell whether it's 1 or 2, and so we select both of them with 50-50 weight. And we kind of keep that information around. And then we remove them, and then the same happens at step 2. We select both 3 and 4 with 50-50 weight. So it's like a fuzzy version of the stepwise selection. And then you can also, it's not just forward, you can go back and iterate. So it also gets around some of the problems. So you might have 2 questions about this. Does this work in practice? And if you're a statistician, you might say this is kind of an algorithmic viewpoint. What's this algorithm actually doing? Will it converge to something? What does it converge to? You could ask that. So let me just give you an illustration to show this can work. Not only can it work, but it can even work in quite tricky situations. So this was taken from a simulation that was chosen so that the causal SNPs here, 1 and 2, do not have the strongest marginal association. So the strongest marginal association is, the y-axis is a minus log 10 p-value for the marginal association. So these guys have the strongest association with y. And the reason is because they're in LD, they're correlated with both SNP1 and SNP2. So they're kind of got a bit of both, and they get a stronger total association because of that. So the intuition is that this is a tricky case because the strongest marginal associations are not causal SNPs, and it's due to LD. Okay, so what happens in our method? Well, here's what happens. What's shown here is the posterior inclusion probability for each SNP, and then they're colored. The colors indicate the credible sets. So there's 2 credible sets identified here, and I'll talk about how we get down from 10 to 2 briefly in a minute. But there are 2 credible sets identified. One contains this causal SNP, and the other contains this causal SNP. And these SNPs here that have the strongest marginal association are not included at all. And the reason, what happens is that in the first iteration of the algorithm, these red SNPs here, which are these strong marginal associations but are false positives, they get big pips. That's what alpha here is. They get big pips in the first iteration because they have the strongest marginal association. But then as the iterative, what happens when iterating is that the second variable gets chosen to be one of these. Sorry, actually one of these, the green ones. Third one, it chooses one of these. It doesn't know which one, but it has these fuzzy weights. And then when it goes back to assess these, it realizes it doesn't really need these anymore because it's got this one and this one. And so in the second iteration, at least, things move towards putting more weight on these guys and less weight on these. And by the 8th iteration, it's really getting down to thinking it needs this SNP and this. And by the iteration 18, the red ones, what's happened here is there are only 2 effects, but we're fitting a model with 3 effects. So what happens is one of the effects, it says, I don't know where to put this. I'll just spread it out across a large number of SNPs. So that's one of the ways that we notice that actually our credible sets are not necessary, is they get extremely big, containing maybe hundreds of variables that are not actually correlated with one another. And we're like, well, that's not very useful. That's not really fine mapping, and we filter those out. Okay. Simulations, you can see the paper for simulations. I'll just say that these credible sets here in simulations have pretty good coverage. These are nominal 95% coverage, and the red ones are SuSiE, and they're varying between 92% and 97% coverage. So that is, most of the sets that we're finding contain a real causal variable, which is what we want. The blue ones is a competing method called DAPG, which has slightly lower coverage, also not too bad. And computational speed, you can see this approach is extremely fast relative to these methods, that other fine mapping methods that are doing some kind of enumeration of all the possibilities. So Caviar is essentially looking at all subsets of size, I think 3 in this case. So it's enumerating all the possible models which contain 3 causal SNPs. I think I'll make this the last slide so I can take questions. So what's this algorithm I've kind of described actually doing? It turns out that it's actually optimizing, or can be viewed as optimizing, what's called a variational approximation to the posterior distribution. So essentially what that says is that every iteration of this algorithm produces a better approximation to the posterior distribution under the SuSiE model. And this, so every iteration is guaranteed to improve the approximation, which essentially means the algorithm is optimizing this objective function and is going to converge. So, you know, it's just an algorithm. If you just come up with an algorithm off the top of your head, they're not guaranteed to converge to anything. But what we can do here is say both that it will converge, and we can say what it's going to converge to, which is an approximation to the posterior distribution. I'll just finish with this example that I showed you at the very start. What happens when we apply SuSiE to this case? We find one credible set, that is, all this red here could be explained by a single causal SNP. It contains, I think, 5 causal variants that are in R squared or R, maybe it's R, according to the figure it's R, of at least 98.7% with one another. So these are in very high LD with one another, so high that it's hard to say which one is the causal SNP. But we think we can narrow, you know, all this red down to a statement that it's probably one of these 5, and then you don't need anything else, in fact. So those data are consistent with a single causal SNP, we think. I'm going to finish so that I can answer questions. We have a paper and software, and I'm happy to take questions. Thanks.
Thank you so much. So there's a question in the chat that I can see. Xiao Wei, do you want to go ahead?
Sure. Thanks, Matthew. So I'm just wondering, for the real data analysis, if I don't know how sparse is the, the structure, how can I make the choice of the L?
Yes, great. So we have 2 ways for choosing L. So firstly, I'll just give you the bottom line, which is it's very robust to choice of L, provided L is at least as big as the number of causal SNPs. So if if there are 10 and you assume there are 2, it's not going to find 10, right? But if there are 2 and you assume there might be up to 10, it will tend to find the 2. And then what will it do with the others? You might think it would find false positives, but what actually happens is that it doesn't know where to put them because there aren't any, right? So what it does is produce these credible sets that span, literally hundreds out of thousands of variables. And these credible sets just contain very large numbers of variables, and they're not these variables are not correlated with one another. So one strategy is simply to prune out these sets that either are very big or maybe more rationally contain uncorrelated variables, because if a set contains uncorrelated variables, it's probably just garbage. And certainly it's not really fine mapping anymore when it contains almost all the... So what we say is in the paper is, well, those sets may or may not contain real causal variables, but they're pretty useless to you whether they do or not. If I give you a set of 100 SNPs and say, might be one of these, don't know which one, and they're not correlated with one another, it's not even really a useful, actionable statement. So we say we discard them. So that's one way. And then there's a slightly more sophisticated way, which is actually we prefer, which is to estimate to introduce a parameter here for each effect, which captures the effect size. And what happens then is that when this parameter here, this is a prior on the effect size for the Lth effect. So there's a separate parameter for each L's. What happens here is if the data don't need an effect, it estimates this to be 0. And so it kind of this is related to something called automatic relevance determination in the literature. It's a trick that can kind of eliminate some of the redundant L's if they're not needed. So there are two ways to deal with it, and with that combination of approaches, it's very robust to choice of L as long as you choose it big enough.
I see. Thank you.
You bet.
I've got a question from Simon.
Oh, yes. Hi, Matthew.
Hey.
I'm sure you get this question all the time, but given that you can find map individual loci very quickly, have you tried applying the similar method and not try to get these printable sets, but just generating polygenic risk scores genome-wide? And does this work better than standard PRS approaches?
So we haven't done it. I mean, PRS is a big, and highly active field on its own, and the clear benefit here is the selection and the uncertainty in the selection rather than necessarily improved predictions. So we preferred to focus on that. I think it would be kind of competitive, but probably not quite as good as the very best methods, because you can, as the polygenic risk score suggests, you can get something out of the polygenic effects, right? And this is a sparse model without it, it's not really designed. It's designed to identify the effects that are big enough to be individually identified and to kind of not worry about the polygenic effects. And you could imagine adding a polygenic component to this model, which we have discussed doing. I think we even did kind of try it, but we haven't getting into the polygenic risk score field and investing our time and effort to do those comparisons is not something we've done yet. Could be done. You're right. So just to be clear, you could add a polygenic, your y equals xb plus a polygenic component plus e, and we have not only talked about doing that, we've kind of done it, I think, but we haven't really pushed it very far.
Would there be a benefit to bringing in annotation information into SuSiE? Great
question. Yeah, so it's easy to do that if you are willing to say how to. So this gamma here is a set of indicators. This is the indicator vector for which one is non-zero, and this pi is the prior probability for each SNP being the non-zero effect. And so you could let pi depend on annotations, and we have done that. And in fact, I think Omar Weisbrod has a paper from Alkes's Price group looking at that and doing that. I can't remember how. You have to have a way of estimating how do you change pi, the prior probability, according to the annotations. And there are several different ways of potentially doing that already out there. And so you could plug, once you've done that, you could plug that in here, and we've done that kind of thing. I guess I'm a little bit I tend to think that it's a second order effect, that the prior, that the annotations don't change your prior by, say, factors of 10 usually, but less than that. And so I kind of would hope that the results wouldn't change that much. And when annotations have a big effect, I'm sometimes worried that people are overstating the importance of the annotations, maybe. But it can be done, yeah. And it has been done. As I think Weisbrod has a paper where they do it.
So I have a question. Hello, hi Matthew.
Hi.
So great talk. Thank you very much. So so you mentioned that you estimate the hyperparameters like sigma square and sigma square sub zero by empirical base, sort of this M step when you're maximizing the evidence lower bound.
Yes.
Whereas in your earlier work in say 2012, you actually do a Markov chain Monte Carlo for sampling those hyperparameters. So I'm just wondering, how robust the point estimate in this framework are in terms of estimating these hyperparameters, and how sensitive the model is to these hyperparameters.
Yeah, in the 2012, are you talking about the Carbonetto and Stevens?
All right.
Yeah, we used importance sampling, not Markov chain Monte Carlo, but I think. But we have stopped doing that. It's a lot simpler to do the maximization, and I think it works generally just as well or better. Yeah. So what can go wrong, I guess, is a question. I mean, obviously, if you're interested in, say, the uncertainty in those hyperparameters, if you're interested in heritability, for example, then it's not really enough to do point estimates for them. But that's not our goal here. So I'd say I'm pretty convinced they're good enough for our purposes here. Without, yeah, and it's my first, it's my default approach now in these kinds of settings is to estimate hyperparameters by optimizing over them, assuming that's easy to do, rather than worrying about their uncertainty as much as I did in previous work.
Okay, thank you.
You bet.
Any other questions? I think maybe we'll take one more. Oh yeah, somebody in QLS, walk up. Yeah, go right ahead.
I have another question. My question is, so you find these credible sets. What do they tend to be? Are they transcription factor binding sites, repressor binding sites, like activator helper binding sites, or are they other things? Have you ever looked at a piece of DNA sequence that you know? Well,
yeah, in the paper we did do a kind of quick assessment of which SNPs are being identified by this. Which SNPs have high pips essentially. What's causing and what kinds of annotations, as Celia kind of brought up, are associated with having a high pip or being in the CS. However, we haven't really done a serious job of doing it, and that's because we decided to focus our efforts on the multivariate case, because I think that we're going to get some really interesting results when we start looking at fine mapping across multiple traits or multiple tissues. Whereas the annotation questions have kind of been done quite a lot with existing methods, and probably the existing methods are good enough for the univariate case. That the existing methods are good enough that people have already gotten some good enough answers in that realm. I mean, and the answer is these SNPs tend to, I mean, it's going to be different for every disease, but you see all sorts, right? You see SNPs where you can say it looks like a splicing factor or disrupting a splicing site. You can see SNPs where it looks like it's interrupting a transcription factor binding sites. You see SNPs in exons, but you also see SNPs where you have no idea why they seem to be associated. So the answer is basically we haven't done a careful, although there's a bit in the paper, we haven't done a careful job of it or we haven't pushed it, because I think that the big benefit of this work is in terms of findings is likely to be when we've got the multivariate case working.
Well, I looking at the time here, and I'm thinking that we really need to wrap up. I actually, though, really want to know the answer to the last question in the chat, which is
about
population stratification and credible sets.
Well, so yeah, I think the short answer, we haven't looked carefully, but the population stratification tends to have really, really big effects on estimates of heritability, for example, because it has a small impact on lots and lots of things. So you have this real difficulty of saying, are these overall inflations across the genome due to population stratification, or cryptic relatedness, or are they due to heritability, polygenic components? And that's you know, LD score regression is one way to try to decompose those, but in general, that's a really tricky problem. Whereas we're just trying to identify the very strongest identifiable effects in a region, and I don't think most of those end up being due to stratification. There could be some exceptions, but I think in your typical GWAS that's on a relatively homogeneous population, stratification doesn't tend to cause, you know, a big effect at a big apparent effect at one SNP. You know, people hold up lactase as an example, for example, but you know, I don't think that it happens very often.
With that, I will express all of our thanks for a wonderful talk. This was really accessible to the students. Thank you so much. You walked them through all kinds of important concepts.